Okay, students, so this is microbiology and chemotherapy, and the course code is CAM 445, and this is lecture 6, and the topic is production of penicillin by the process of fermentation, part 1, and I am Dr. Saeed Ibrahim, Assistant Professor, Department of Chemistry, Biochemistry of Science and Technology, and you can access all these lectures through the KCMS account, where you can mark your attendance, and then you can get the link from there, and then you can access it through the ms.cohort.edu.pk media server of KUST. Now, the topics which we will discuss in this lecture, they include the production of penicillin, antibiotics, mode of action of antibiotics, and methods for antibiotic production. Now, for the production of penicillin, what are the learning objectives for production of penicillin? That as a result of discussing or studying the production of penicillin, what you will get to understand. So first of all, you will be able to identify the useful products from microorganisms. So in the coming slides, you will come to know that how the microorganisms, they are producing the primary metabolites and the secondary metabolites, and the secondary metabolites are beneficial, and then those secondary metabolites can be utilized as antibiotics. Also, to identify the microorganisms which are used and the main stages in the production of penicillin. So that is also very important, that what steps need to be taken when the production of penicillin is carried out. And obviously here we will be talking about the production of penicillin through fermentation, and also to be able to describe how downstream processing is carried out to extract and purify the end product of fermentation. So some of the part of the downstream processing will be discussed here, and some of the downstream processing part will be discussed in the next lecture, to clarify that how the steps are being taken so that the end product of fermentation is in highly pure form. Now, moving towards antibiotics. So antibiotics are antimicrobial agents which are produced naturally by other microbes. Normally the antibiotics, they are produced by fungi or bacteria in terms of natural sources. The antibiotics are also produced or synthesized synthetically, but they are more costly in comparison to the production of antibiotics by using the fermentation technology. Now, the important thing here is that the microorganisms, they are producing the primary metabolites and secondary metabolites in their metabolism, and the secondary metabolites, most of them are beneficial and some of them are antibiotics. So if the process of fermentation is manipulated in such a manner so that the microorganisms, they are producing more of the secondary metabolites, so then as a result of purifying them, isolating them, extracting them, the antibiotics in pure form can be obtained, and then they can be used for treating different diseases. Now, the first antibiotic was discovered in 1896 by Ernest Duquesne, and further it was rediscovered by Alexander Fleming in 1928 from the filamentous fungus, which is known as the Penicillium notatum. Now, moving towards the mode of action of antibiotics. So, antibiotics can be selectively toxic by targeting such features as the bacterial cell wall, the 70S ribosomal portion, and the enzymes that are specific to bacteria. Furthermore, in this way, the human eukaryotic cells are unaffected. So here we need to understand that when we are taking the antibiotics by mouth or by injection, so what is happening is that the antibiotics, they are targeting the bacterial entities. So for example, the 70S ribosomes and different enzymes which are having a very important role in the metabolism of the bacteria. So this is a good thing and a beneficial thing for the human beings, that the antibiotics are not attacking the human cells. Um, so in that case, only the bacterial infection is being treated, because the antibiotics, they are attacking the bacterial cells and their metabolic reactions, the ribosomes. So as the ribosomes are integral part of the bacterial well-being, so if the ribosomes are being attacked, so it means that there will be no protein synthesis taking place, and as a result there will be no growth and ultimately no reproduction of the bacteria. So other antibiotics may affect, for example, the cell membrane of the bacteria. So when the cell membrane of the bacteria is being targeted, and ultimately it is being damaged or destroyed, so the bacterial cell is no more intact, and it cannot perform its normal activities because the cell membrane is damaged. Some of the antibiotics, they are attacking the DNA replication process. So DNA replication process is very important process in the life cycle of the bacterial cells, and if there is no DNA replication taking place, so it means that the DNA quantity will decrease, and as a result the translation as well as the transcription processes cannot take place, and ultimately the protein synthesis will stop. So that's why the DNA replication process is being attacked by some of the antibiotics, whose mode of action is to attack certain enzymes who are responsible for the DNA replication. Some of the antibiotics, they are also attacking the transcription process. So the transcription process is the conversion of DNA into RNA. So, and they are only attacking the enzymes uh, responsible for the process of transcription within the bacterial cell. So again, this is important to note that the antibiotics, they are attacking the cell membrane or the DNA or the RNA of the bacterial cell, because the composition, the structure of the bacterial DNA, bacterial RNA, and cell membrane of the bacterial cells are different from the human cells. Another process is the translation. which is the conversion of RNA into protein. So again, here the ribosomes and the RNA, messenger RNA, transfer RNA, ribosomal RNA are involved, which are being attacked by the antibiotics, and as a result, the conversion of RNA into protein is not possible, and as a result, no growth and reproduction of the bacterial cell is possible. Now, moving towards the examples of the antibiotics which are attacking the bacterial cells at different stages of their life cycle and of different organelles in the bacteria. So, examples are penicillin, ampicillin, amoxicillin, methicillin, and what is their function is that they inhibit the enzymes which are involved in the synthesis of peptidoglycan, which is present in the cell wall of cell membrane of the bacteria, the cell wall of the bacteria, and as a result they cause cell lysis. So when the cell have been lysed, so there is no concept that the bacterial cell can maintain itself to reproduce or to grow. Some of the antibiotics, they are bactericidal. So there are two types of antibiotics. One are bactericidal, which means that they kill the bacteria, and the other ones are bacteriostatic, which means that those antibiotics, they stop the growth of the bacterial cell. Now, some of the antibiotics, they are narrow spectrum, uh, so they have little effect on gram-negative bacterial cells. So some of the antibiotics, they are broad spectrum antibiotics, so it means that they can work on a number of species of bacterial cells, like gram-negative bacteria as well as gram-positive bacteria. And some of the antibiotics, they are narrow spectrum, which means that they are effective on gram-negative bacterial cells, but they are not effective against gram-positive bacterial cells. Now moving towards the penicillin, so it is the antibiotic substance named penicillin, and it was not purified until the 1940s by Flore and Chain, and it was just discovered and just in time to be used at the end of the Second World War. So it is important to note that during the Second World War there was no antibiotic available to treat the soldiers, but during the end of the uh, Second World War, around 1943-1944, the penicillin was available, as was purified by Florian Chain, and then it was produced in huge amounts by the process of fermentation, and then purified, and then used on soldiers who were suffering from different types of infections. Now, penicillin was the first important commercial product which was produced by aerobic submerged fermentation. So it means that the process of fermentation was carried out in the presence of air, and also the penicillin was produced by the process of fermentation, and not by the process of synthetic chemistry. So it is important to note that the penicillin was the first antibiotic which was produced in the history of science, and it was a very major success, considered to be at the end of the Second World War. So The penicillin, when the penicillin was first made at the end of the Second World War, and it was produced by using the fungus known as Penicillium notatum, and the process made 1 milligram per decimeter cube. So you can see that from 1 liter of the fermentation uh, culture, only 1 milligram of the penicillin was produced. So at the time of the Second World War, as there was a high need of the antibiotic penicillin, and you can assume that the quantity or the percent yield of the penicillin was very low at that time, and a lot of fermentation tanks were being utilized so that to produce maximum amount of penicillin so that it may be available to the soldiers for treating their infections. Nowadays, in current situation, a different species is used which is called Penicillium crassugenium, and along with it a better extraction procedures is used. So the yield is now 50 gram per decimeter cube or 50 gram per liter. So from here you can assess that through the technology, through the better extraction process, the yield has been increased up to 50 gram per liter from a fermentation process. So the important thing is Penicillium crassugenium, which species is used of the penicillium, as well as the extraction procedure. And there's a constant search for improved yield. Right, so moving towards antibiotic production. So there are over 10,000 different antibiotics which are known in the scientific community, but only 200 are in commercial use. So it means that the researchers, they are working and analyzing the drug efficacy of different types of compounds, and they have discovered around 10,000 antibiotics. Now why only 200 are in commercial use is the reason that there is a long process between the discovery of an antibiotic and finally uh, its availability for the commercial use. And these steps include toxic studies, animal trials, clinical trials, which take a long time. So that's why only 200 antibiotics have made it through the commercial use. Now, it has been observed that the new antibiotics are no better than the previous ones. So the reason for this is that day by day the research is going on, and the new antibiotics are being discovered, and then after a long thorough process they are available for the general public. But it has been observed that the new antibiotics are not very much effective in comparison to the existing antibiotics. And the reason is that as new antibiotics are introduced, so the bacteria, they develop resistance against those antibiotics with the passage of time. Now there is also a constant search for new antibiotics in the pharmaceutical industry, and also the antibiotics, these are the most prescribed drugs and are big business. So, in common examples, if you think about antibiotics like meltine, ciprofloxacin, levofloxacin, and other antibiotics. So, whenever you have a slight fever or you are suffering from chest infection or you have sore throat, so when you visit doctor, he is normally prescribing antibiotics along with the other drugs. Now, finding a new antibiotic and getting it into the market is a very long process, as I discussed it earlier, and it can take up to 15 years. So you can see that it is a long time, after which the antibiotic is being tested, it is being processed, it is being analyzed, and then after all those tests and trials and clinical trials, animal trials, and trials on volunteer patients, ultimately it gets to the market, and it can take a maximum of 15 years. Now these are the references for further reading and understanding. So the first one is 11 centrifuge reviews of medical microbiology and immunology. The second one is Carson, Basic and Clinical Pharmacology by McGraw-Hill and Company. And the third one is Lippincott Pharmacology. The fourth one is Cell Biology by R. Taubin. And the last one is Fundamental Principles of Bacteriology by A.J. Seller. So related to this lecture, the first one, reviews of medical microbiology and immunology, and the fifth book, which is Fundamental Principles of Bacteriology. So these are good books which you can read further for understanding and clearing your concept. Thank you.